Good afternoon, everyone. It gives me great pleasure to present the progress made by the Translational Biomarkers of Neurotoxicity, or NeuTox for short, committee since its inception in 2012. And I'm presenting on behalf of the three of us who co-chair this, David Caligaro and Merle Paul, who are hopefully also on the line. I heard Merle dial in. So if I could go to the next slide, please, Jen. Great. So at a glance, and we'll go into a little bit more detail on each of these points as we progress. Our inception was in 2012 as an Emerging Issues Committee, and since then we've grown considerably, and over the last 3 years as a full committee, we've grown a lot. So our membership is currently 15 industry, 8 government and 11 academics. We have, in summary, really done a great job with our publications so far, with 2 publications accepted and a 3rd in draft. And we'll say a little bit about that in a minute, a bit more about those in a minute. But notably, the first publication, which went into top sci, came out as in their top 10 citations list for 2016, which was excellent outcome for us and really great to see that. We're partnering with the HESI Genomics Committee, with the National Centre for the Replacement, Refinement and Reduction of Animal Usage, NC3R, based in the UK. We're partnering with IMI and with the Japanese Consortium for Safety Assessment using Human iPS cells, or KSAHI. We've also so far made 5 presentations at international scientific meetings. We have 1 and now actually 2 scheduled for 2018, 1 at SOT and 1 at the Experimental Biomedicine meeting in San Diego in April. So that's the committee at a glance, looking at the timelines of where we started and where we're up to. Next slide, please. So history, we were voted through on an emerging issues proposal in 2012. We convened a workshop at SOT in 2013 to really scope out what it was that we were trying to do. We brought in a huge range of experts, everyone who had an opinion and an interest and something to contribute to that workshop, which was a great session where we really brainstormed everything about the topic. Based on that, we came out with a clear plan, which we wrote up initially as a white paper for our own use, but then we realised it was the basis of a good quality publication. So we prepared that and submitted it to ToxSci, and it was published in 2014. The purpose of getting that published was twofold. One was really to make sure everyone knew what we were doing, but secondly, that white paper was a statement of purpose and intention, but also set out in quite a lot of detail the experimental protocol that we were planning to use to test the hypotheses that we had to address. So others were able to comment on that publication and contribute their expertise if they had views on what we were planning to do. So we set out the protocol in terms of species, neurotoxicants, time points, biomarkers to look at in a paper. And as I say, that was on the top 10 list in 2016 for ToxSci, which is great. Based on that, we then progressed, and during 2015, we ran the pilot study using trimethyl-tin in the rat, and that was a multi-centre effort, but done primarily at the FDA and CTR, and we'll hear a little bit about that later. But many people contributed to that, both in terms of intellectual and sweat equity with experimental work and so on. Another point that's key is in 2016, we added in a sub-project, microelectrode array, because as we were working through our data and through the concepts, we realised that the ability to perhaps address seizurogenic risk in vitro using microelectrode array was another key endpoint of relevance to this project. And we'll say again a little bit about that later on. In 2017, the data from the pilot study with trimethyl-tin are due to be published. In fact, that paper's just been accepted and is an EPUB ahead of print. I guess it may come out in paper in January. So really nice paper, and you'll see that a little bit later on. And now we're planning for phase 2, which is to really push on the microelectrode array, and also our phase 2 studies following up on what we've achieved so far with phase 1. Next slide, please. So the committee is co-chaired by Merle Paul, USFDA, David Caligaro from Lilly, and me from the University of Birmingham. And we've co-chaired this 3-way since its inception, which has been a really great team. We are guided, assisted, and managed by Jennifer Pearson and Alex Feitel, which is a great team to help us stay on the straight and narrow and keep track of all the information and data. So this is the leadership, and any questions, obviously, about this can be addressed to any of us. Next slide, please. So looking at membership, as you can see from this, our current membership is extensive and diverse. It includes public, private sector from the US, from the UK, from Japan, from Europe, a significant number of universities, pharmaceutical companies, not-for-profits, and small companies included in this. And all of our members are really contributing extensively from their different perspectives, both from the academic discovery point of view, but also from the regulatory implications of some of the work that we're doing, and the more applied from the pharma industry, so how can this be used? So it's a really extensive and diverse team of contributors and membership, which has really enhanced and enriched what we've been able to do so far, and what we hope to do in the next few years. Next slide, please. So there are 2 missions. The first one is to identify biomarkers for improving the prediction of neurotoxicity. So this is obviously the start, what we started with as our mission. And just a little bit of retrospective, as many of you will be aware, is that it's extremely difficult to predict neurotoxicity in all aspects of toxicology, whether it's drug development, environmental exposure, and so on, or chemicals, agrochemicals. And certainly from a pharma industry point of view, we really rarely discover neurotoxicity until it's too late in a drug program. So it would be it's a really key objective and tremendously helpful to be able to predict earlier, both in time or earlier, both in terms of further down the dose response curve, the potential for a new drug or a new chemical or an environmental toxicant to cause neurotoxicity, other than having to wait for the ultimate invasive pathology that is often required, or behavioural abnormality that can sometimes be seen in toxicology studies. So that's our aim, and that has been a key aim from the start, and remains very much an absolute vital step in neurotox, is improved biomarkers. As I mentioned earlier, as the project has evolved, we've added in a second mission, which is using microelectrode array to identify seizure-genic compounds in vitro. Again, this will be tremendously valuable to assess the potential for seizure in vitro without having to go into animal models, and it's tremendously important for new drug development, but also for assessing environmental chemicals and exposure in the environment. So we have 2 key missions. Next slide, please. These overlap extensively in that they both are aimed at improved translation of what we see in vitro to in vivo, and what we see in animals in vivo to what we see in humans in vivo. So that we're looking for improved translation in vitro, in vivo to humans. They're minimally invasive, such that the endpoint can be assessed without any invasive procedures, and can be assessed in life with some of the endpoints. They aimed at giving earlier decision making, and also de-risking drug projects earlier and prioritising projects, but also around decision making, de-risking, and prioritisation of chemicals for risk assessment. Both of these are also aimed at generating increased mechanistic understanding of neurotoxicity in all its different aspects. And of course, if we're successful, as we intend to be, both of these approaches, biomarkers and NEA, will result in a significant reduction in animal use in assessing the endpoints of neurotoxicology. Okay, next slide, please. So the pilot study I mentioned earlier that took place during 2015, and the data are just literally in pre-publication at the moment. The study aim was to investigate circulating biomarkers that predict central and peripheral neurotoxicity resulting from exposure to trimethyl-tin. And for those of you who recall the details of this in the white paper, trimethyl-tin was chosen as an example neurotoxicant because it's well understood in terms of pathology, but it's also its main, it is very effective at causing neurotoxicity in the rat, which after all is the main tox species used in many aspects of regulatory toxicity testing. There are obviously other neurotoxicants that are used mainly in mouse models, but on balance, after a lot of discussion, the expert group chose TMT in the rat as our model. That went ahead with different time points. Data analysis was completed. We collected additional samples for metabolomic and lipidomic analysis. And as I've mentioned, the first manuscript is completed, and you'll hear a little bit about that in a moment, and more manuscripts are in progress. So the manuscript that's just was submitted in August, which contained all of these data, is in press now, and I believe will be in paper press January. So in the next few slides, I was going to hand over to Saeed to take us through the details of the results, the protocol and the results. Unfortunately, Saeed is on the line, but he has laryngitis and he can't really speak. So Jennifer has kindly volunteered to step in for Saeed and take us through the next 4 slides of data, and then hand back to me to conclude from slide 13 onwards. So again, Saeed, if we need your help, we may need you to chip in if we've misstated anything or we've overlooked anything key. But I'm sure Jennifer is on top of the data and can present it. So over to you, Jennifer, please.
Great, thanks, Ruth. Yeah, Saeed, you can save your voice for all the hard questions. So as Ruth said, that this was our first pilot study, and we really wanted to identify circulating biomarkers in bodily fluids and tissues that were associated with neurotoxicity. And so not having really any known fluidic biomarkers, we wanted to ensure that we had complete tissue samples collected: brain, liver, thymus, the adrenal system, kidney, spinal cord, sciatic nerve. A whole host of other tissues were also collected, but I don't think those were listed since they didn't turn up any results, in addition to all of the fluidic CSF, plasma, serum, and urine. And so in addition to these, we also looked at traditional behavioral endpoints and histology to be able to compare back and forth. So looking just at kind of a snapshot of what we looked at in this study, from the brain, we wanted to look at oxidative stress, and we looked also at the hippocampus. And so some of the main tissues and proteins that we were looking for included synapsin, fluorojade, FJ, GFAP, and that GFAP is actually a really common protein that's looked for in the brain to do any type of neurotox analysis, and then TGF. And so we found that oxidative stress was in fact increased as we expected it would be within the brain and within those tissues, and so that was good to see something that we would expect. And then also from the cerebrospinal fluid, the CSF, we saw a similar increase in the oxidative stress, so that was a really good correlation there. And then moving into the serum and plasma, we looked at carnitines, lipids, and the metabolome. The metabolome analysis is still ongoing. It's been a little bit complicated, and so we're hoping to be able to include more of the results of that in the next paper. And then we also looked at urine. And so we saw some really good correlation between all 3 of the fluids that we took. And specifically, you can see in the TGF was in all 3. And so we are hypothesizing that the brain is releasing these biomarkers and it's crossing the blood-brain barrier and entering into the urine and serum, and so that we're able to see that correlation. So I think we'll go into a little bit more detail of some of the results in a minute, but I think just in general that, you know, some of these markers here are things that we'll want to focus on in our next study and see if they continue to correlate. So this is a full summary of the pilot study that we saw. So we knew going into this, after kind of an exhaustive list of other different compounds that we could have used, that TMT was really going to be kind of a hammer of neurotoxicity. We knew what we would get in the brain, and so we knew that if there was something that would cross the blood-brain barrier, we would see it with TMT. And so the final dose that we ended up using was 7 mg per kg, and that was IP dosed. I think we had started with 8, but animals were dying too soon, so we needed to dial it back a bit. And then we collected samples at 2, 6, 10, and 14 days so that we could see the time course of the TMT. And we did, in fact, see some recovery by the 14th day in some of our markers. And so, again, as I said, we did the behavior study to just compare to what's generally done with neurotox. And so there we did see and expected to see the reduced locomotor activity, and that was observed after day 6 of dosing. We also did an imaging analysis using MRI, and so that revealed that there was an impact on ventricle size, and so that is something that's ongoing. I think the folks at NCTR have been doing these MRI studies for a while, and so it'll be interesting to be able to compare to some of the results that they had seen in the past compared to this study as well. And so, as I had mentioned, that we did see a time-dependent change, specifically using the GFAP, the fluorojade C staining, and the IBA1. And some of that, I think, particularly the GFAP, we did see some recovery towards the end, which we're not sure if it's recovery or if the cells are just dying, so we're not picking it up anymore. So that's another something that we want to try and explore a little bit further in a future study. So we did see the protein changes in both the serum and plasma, and that correlated with the brain damage in the synapsin and the GFAP. And then again, we saw the TGF beta appeared to be a powerful biomarker, and we saw the changes that correlated with both brain and urine. And so that's one that we've kind of flagged with some specific interest for the next study as well, and hoping to be able to use that to kind of further show some correlations between brain and body fluids. So the metabolism or the metabolome analysis that we have done, we did see that there were alterations in several different metabolites. And so that's ongoing, and we hope to be able to show that there's even more correlation there. We also did an miRNA analysis, and we worked closely with the HESI Genomics Committee and with Lilly to do this analysis, since they have a lot of experience with the mRNAs. And so there were 35 brain-specific mRNAs that we looked at, and many of these showed significant changes in both the CSF and in serum. And so the ones that showed significant changes, again, will be flagged for a future study so that we can really determine where the correlation lies there. So we also are working on a proteomics analysis. We did see multiple changes there within proteins in the CSF, and so we'll continue to look at that. And hopefully we'll be able to display those results in the third paper. And again, we did histopathology just to ensure that what we were seeing was correlating. So we saw some mild kidney damage in some of the TNT cases, which is not to be unexpected. But we do want to make sure that what we're seeing with the kidney damage isn't what's causing the TGF-beta biomarker to appear in urine. But I think looking at, without having done a complete statistical analysis on this, looking at the number of animals that had kidney damage versus the number of animals that we saw the TGF-beta increasing in, that we probably will be able to kind of rule out that it was kidney damage related, showing that biomarker to increase. So overall, you can see that we had quite a few increases or decreases in a lot of these different proteins here. And so a lot of the statistics have been completed and are still ongoing, but P was set to 0.05. And looking at the increases with all the different proteins and markers, clearly there was quite a few more of those. So I think that this is where we're really going to be focusing on for our next study with all of these different markers here on the right-hand side of the screen. I don't know if you can actually see my mouse. I'm trying to point to things, but that's really where on the right-hand side of the screen where we saw the increases. I think that will help us to better understand where the blood-brain barrier changes are occurring and hopefully help us to really narrow down a useful suite of fluidic biomarkers. So I'll stop there and see, I don't know, Saeed, if you wanted to try and add anything else or if we can turn it back over to Ruth.
I think we can just go back. I think you did a great job.
Okay, thanks. So I'll move on and turn it back to Ruth.
Thanks, Jennifer. Thanks, Saeed. So just reiterating what Jennifer explained in the last slide, we're tremendously excited about the fact that we see biomarkers in cerebral spinal fluid, which means that they're clearly coming from the brain, but several of those are then reflected into the serum, which, as I'm sure you appreciate, is a much less invasive and a much easier way to monitor neurotoxicity. So we're really excited about seeing all these things correlate together, and it gives us a tremendous route forward for further exploration. So thanks very much to everyone who did those experiments and contributed at Lilly and FDA and CTR. There's a tremendous job on all that experimental work. Okay, moving on. The microelectrode array project has evolved out of the original NeuTox, or Biomarkers of Neurotoxicity project. And we now have named co-chairs of Daniel Millard from Axion, Tim Schaefer from EPA, and Chris Strock from Cyprotex. These teams working together on a retrospective of data analysis, and they're sharing data via webinars, collating data and refining their plans for a pilot study to evaluate the hypothesis that microelectrode array is a powerful tool to assess seizurogenic potential in vitro. There's a pilot study planned with 10 toxins where there are known seizurogenic effects either in the clinic or in preclinical animal models. So we're planning to scope that out with these 10 toxins in vitro. At the moment, we're working on the protocol and the parameters by sharing previous study protocols and parameters between the different participants, such that we can bring together an overall protocol that everyone can contribute to and gives us the very, very best science based on the state of the art. I'm sure those of you who follow this field know that there are many different methodologies and approaches to MEA, and what we're about in this scoping phase is to ensure that we evaluate those where the science is strongest behind them. So that's undergoing at the moment with many ongoing webinars where we're sharing data between us and to plan for this. Okay, so this is growing. The partner interest continues to grow. 10 new members joined us in 2017, which is great, and that's progressing really, really well. Next slide, please. So the Fluidic Biomarkers Project, as we said, we have one paper in press and just about to be published. We have a symposium submitted for consideration for SOT in 2018. So that's all ongoing at the moment in terms of dissemination. We're planning for the phase 2 study to explore potential biomarkers of neurotoxicity based on what we've seen so far, taking a real deep dive into the data, deciding what questions remain to be answered and which further we can take this. The other really exciting development that's emerged in the last few weeks that we do need to explore, and certainly not a done deal, but to explore how relevant what we've found is for other types of neurological injury, particularly Parkinson's or perhaps even CTE or chronic traumatic encephalopathy. If we think about it, one of the major challenges in developing new drugs for Parkinson's is that we do the toxicology data to support a 1-month clinical trial. But 1 month is not sufficient really to be showing any improved and often an improvement in Parkinson type endpoints in the clinic. But unless we have the toxicology to support a longer exposure in the clinic, then that can't proceed. If we were able to develop biomarkers of the onset of Parkinson's, or indeed cessation of the symptoms of Parkinson's, even though symptoms have not yet been ameliorated, that would be incredibly powerful in aiding drug development for endpoints like Parkinson's. But we can also see tremendous opportunity to extend what we found to CTE as well, which is obviously a big issue in sports and in the media at the moment. So we see great potential to scope and take this beyond where we started to really maximise the impact of the work we've done. At IMI2, we've been talking to the IMI2 folks extensively over the last 6 months with a view to a collaboration that will really extend our reach and reach into the clinic. And that's been a really good collaboration and synergy between us and IMI2 to bridge and improve our translatability between in vivo to a clinical link. And then the other part, of course, is the mitral electrode array project. The pilot study is just about to start, and we're expecting results next year, depending on how long it takes, and then obviously data analysis would come after that. Okay, so those are the scientific results, progress and data.